Good morning, students. In our last class, we had discussed about fertilization. Fertilization occurs by double fertilization. We saw as a result of double fertilization, the zygote had formed and the embryo, the zygote and the endosperm had formed. Today we will learn about the post-fertilization events. Post-fertilization events occurs after fertilization are the events which are included or which happens after fertilization. They include endosperm development of the endosperm. The first step is endosperm development, second one is embryo development, then comes your formation of seeds from the ovule and formation of fruits from the ovary. So these are the various steps that take place in the post fertilization. In after fertilization, it begins with endosperm development, then embryo development, and after this the seeds will develop, the ovule will develop into seeds and ovary will develop into fruits. Now let us begin with endosperm development. Endosperm development precedes embryo development. It precedes embryo development. What do you mean by precede? Precede means before. It happens before. So it's all, in most of the cases, in most of the flowering plants, endosperm development will occur before embryo development starts. The purpose is to ensure that there is sufficient nutrients for the embryo development. Once the embryo starts developing, there will be a need for a large amount of nutrients. In order that the plant supplies this, the endosperm development will happen before. What is the purpose of endosperm? To provide nourishment to the developing embryo. So this has to happen before in order to ensure that there is always sufficient food reserve. There is sufficient food reserve for the embryo development. So what happens here is, the primary endosperm cell, I hope you remember, the central cell after double fertilization develops into the primary endosperm cell. This develops to form the triploid endosperm tissue. Triploid endosperm tissue. I hope you remember why it is called triploid. We had seen that the two polar nuclei and the male gamete fuses. Therefore, the primary endosperm nucleus was triploid. The primary endosperm nucleus was triploid. So when the nucleus is triploid, the cell will be also triploid. Therefore, the endosperm tissue will be triploid. In this tissue, there will be a large number of cells. Now, these cells are filled with food materials. These cells are filled with food materials which ensures nutrition to the developing embryo. Which ensures nutrition to the developing embryo. Now coming to the primary endosperm nucleus. The primary endosperm nucleus, that is in the beginning, when endosperm development begins, it will be free nuclear division. It will be free nuclear division. What do you mean by free nuclear division? Free nuclear division means only the nucleus keeps on dividing. Only the nucleus keeps on dividing. So that stage is known as free nuclear endosperm. Free nuclear endosperm. Later only the cell wall formation takes place. Later only the cell wall formation takes place and the endosperm becomes cellular. The endosperm becomes cellular. That means first this is your large central cell with the antipodal cells, the synergates and the zygote here. And here you have the triploid primary endosperm nucleus. This triploid endosperm nucleus will start undergoing free nuclear division. That is, only the nucleus divides. Only the nucleus is dividing. So the division is called as free nuclear division. Free nuclear division. Once again let me tell you that this is the embryo sac. You have the zygote here. You have the triploid nucleus here. So this will undergo, the nucleus will become 2, 2 becomes 4, 4 becomes 8, 8 becomes 16 and so on. The nucleus keeps on dividing. So in the beginning it will be free nuclear division. Only the nucleus divides. That is why it is known as free nuclear stage. It is known as free nuclear stage. After some time, what will happen? After there is sufficient number of nucleus, what will happen is nuclei, cell wall formation begins. Cell wall formation begins. These nucleus will develop cell wall around themselves. When this happens, they come to be known as the cellular endosperm. Cellular endosperm. Hope it's clear. So first the primary endosperm nucleus undergoes free nuclear division and the stage is free nuclear endosperm. Then the cell wall formation takes place and the endosperm becomes cellular endosperm. All of you must have seen tender coconut. Tender coconut. In tender coconut means the telaniru. In that tender coconut, you must have seen water and a soft kernel. Water and soft kernel. Kernel is that soft white part of the tender coconut. This water is the free nuclear endosperm and the kernel is the cellular endosperm. Cellular endosperm. You must have seen that in the beginning, this tender coconut will be having large amount of water and less amount of kernel. With time, what happens? The free nuclear endosperm or the water becomes lesser and lesser and the kernel becomes thicker and thicker. That means the cellular endosperm formation is taking place. Right? You must have seen a normal coconut. In a normal coconut, have you seen a spongy white ball-like structure near the three eyes of the coconut? The three eyes of the coconut are the micropiles. A special case, there will be micropiles. Now, and the micropylar end, you have the developing embryo. What you see, that spongy soft thing is known as actually the embryo, which is getting developed to form a new individual. What is happening during the process of formation of embryo? You find there will be lesser and lesser amount of water, which indicates that the free nuclear endosperm is becoming lesser and the cellular endosperm is taking its place. That's all about endosperm development. Coming to the second part of post-fertilization event, that is embryo development. Embryo development. Embryo development always occurs in the micropylar end of the embryo sac. Micropylar end of the embryo sac. Do you remember this diagram? This is the micropyle. This is the micropyle. So the zygote formation has taken place here. Right? So the zygote is present at the micropylar end. Therefore, the embryo development occurs at the micropylar end. The stages of embryo development is known as embryogeny. Embryogeny means the stages of embryo development. Stages of embryogeny is similar in both monocot and dicot to a great extent. To a great extent, they are similar, but there are differences, but mostly they are similar. Okay, so the first stage of embryogeny is proembryo, then globular embryo, then heart-shaped embryo, and then the mature embryo. Have a look at the diagram in your textbook. So the zygote, it becomes two-celled. 2-cell becomes 4, 4 becomes 8, 8 becomes 16. So it attains a shape of a globular embryo, a globe or a globular embryo. After certain stages of development, it attains the shape of a heart. Therefore, it is known as heart-shaped embryo. Then the next stage of development is known as the mature embryo. So from a single cell zygote to a 2-celled proembryo, then 2 becomes 4, 4 becomes 8. After some time, it attains a shape like a globe and you call it as a globular embryo. Then after certain number of divisions, it attains a shape of a heart, therefore known as heart-shaped embryo. Then after some time, it becomes the mature embryo. Now let us learn the structure of the dicot embryo or the dicotyledonous embryo. The dicotyledonous embryo has, a dicot embryo has an embryonic axis. It has an embryonic axis and 2 cotyledon. An embryonic axis and 2 cotyledon. This is known as the embryonic axis and the 2 cotyledon. What is the role of cotyledons? The role of cotyledons is to store food required for the development of the embryo. Okay, then the part of the embryonic axis, so this is your embryonic axis, the part of the embryonic axis above the cotyledon, that is this part, the part above the cotyledon is known as epicotyle, and the part of the embryonic axis below the cotyledon is known as hypocotyle. So epicotyle and hypocotyle. Epicotyle develops to become the plumule and hypocotyle develops to become the radicle. What does the plumule develop into? The shoot system, and the radicle develops into the root system. The next one is a monocotyledonous embryo or a monocot embryo. As the name suggests, there will be only one cotyledon. Dicot has two cotyledon, monocot has one cotyledon. So there will be an embryonic axis here also, and there will be a cotyledon on the lateral side. So here also this is known as the embryonic axis, and this is known as the cotyledon. But there is another name for this cotyledon in a monocot. In a monocot embryo, it is known as scutellum. 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 Okay, it's also known as the scutellum. The part here also above the cotyledon is known as epicotyle, and below the cotyledon is known as hypocotyle. As usual, epicotyle develops to become the plumule, which develops to become the shoot system. Hypocotyle develops to become the radicle and into the root system. Now, there is a peculiarity here. The epicotyle is covered by some undifferentiated sheet-like structures, which is known as coleoptile. And the radicle or the hypocotyle is also covered by some sheath-like structure which is known as coleorhiza. The role of these two, they are protective layers. They protect the hypocotyle, and the coleorhiza protects the hypocotyle, coleoptile protects the plumule. That's all about your embryo development.